This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 956, recorded on November 17, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 35 Fahrenheit, but it says it feels like 25, and we're going to have light snow starting in 13 minutes. And in Celsius, that's 1 degree, feels like -5 Celsius.
Here in New York, it's 7 degrees Celsius and mostly cloudy. Also joining us from Austin, Texas, Rich Condit.
Hi there. 57 degrees Fahrenheit, cloudy, and it's not going to get much better for a while. We're in the cool phase. I heard that Buffalo got 4 feet of snow. Man.
Wow.
I missed that. Sort of.
We have two guests for you today who worked on a project together that we are going to talk about, but now they are separated. So returning actually from TWiV 935 from Cornell University, Cedric Feschotte. Welcome back.
Thank you. Thank you for having me again.
Sure. Last time...
No, we don't have 4 feet of snow here, even though we are upstate New York, not too far from Buffalo. But we got some. We got a dusting. You know, it's pretty. It feels like winter already. Yes.
So last time I visited you, it was summer, I guess, right?
Mm-hmm.
It was very nice weather.
Yeah.
Went to your office, and we didn't talk too much about your work. So we mostly talked about background. So I told you, when you get something interesting, let me know, and you did. So that's what we're here today for. And also joining us from Yale University, John Anthony Frank. Welcome to TWiV.
Thank you for having me. It's 45 degrees here and overcast. Can't complain.
In New Haven, Connecticut, right?
Yes.
John, where on the Yale campus is your laboratory located?
We're in the Anlian Center, so I work for Dr. Akiko Iwasaki. So we are on College and Cedar, although I get the roads wrong, so I could be a little bit off there. We're next to the York Street Hospital, or closest to that.
Okay.
So, John, you were a graduate student with Cedric when this work was done, is that right?
Yep. He was very kind enough to rescue me from a previous grad lab and allow me to work on this project. So it's been fun to work on it.
How did you get to Cornell? Where were you before that?
I was at the University of Utah at about the same time Cedric was there. And so when I joined the lab, basically I moved along with him from Utah to Cornell.
Okay.
So Were you a graduate student at Utah?
Yes. I started as a graduate student in Utah, and basically I got lucky enough to be able to move with Cedric to Cornell and continue my graduate studies there. Sorry, go ahead.
Are you originally from Utah?
No, I grew up in Germany, actually. I spent 18 years over there and then went to undergraduate at Linfield College in Oregon.
You speak German, I guess, right?
Yeah, although I'm a little bit more reluctant to use it because I've lost quite a bit of it since then.
All right, we won't ask you to say anything.
So what got you interested in science?
I've always kind of just been a curious person and really just enjoyed understanding how things work. It's just kind of always been what I wanted to do. And like many graduate students, I thought I initially wanted to go into medicine and become a doctor. And then quickly realized that doctors actually have a lot of hard things to deal with, especially dealing with patients. And they don't necessarily always listen to the best advice that they get. And so I realized maybe science and science education is maybe more where I want to go.
So where'd you do your, 18. So where'd you do your undergraduate work?
My undergraduate work was in a small liberal arts college at Linfield College. It's really tiny in Oregon. And basically there I studied the function of chordactin proteins. And really what we were trying to figure out there was how do different phosphorylation sites on chordactin, which is this sort of cytoskeletal protein that plays a role in cell cell growth and cell movement. How do those phosphorylation sites impact sort of the movement of Ewing sarcoma cells?
And are you on sort of disciplinarily the same path that you were in Cedric's lab at Yale, or have you changed things around?
I still try to. I work on several different projects. I think the project that's most similar to what Cedric works on is I've been looking at the genome regulatory activities of a restriction factor called ApoBec3A. Although that project has kind of been sitting a little bit, I'm working in tandem with that project on also trying to use evolutionary studies to sort of see if we can, using the evolutionary history of species, figure out maybe which species might be harboring viruses that could be reservoirs for new zoonotic transmissions. Basically asking where is their evolution going on, sort of host lineages, and then asking in those lineages, are any of those receptors similar enough to humans that could maybe move between species? Okay. So I try to take a little bit of the concepts that I learned from Cedric to sort of apply it to my own work.
So what are you going to do if you grow up?
Great question. Ideally, I would love to teach. That's really sort of what I want to do. So I've tried to tailor sort of my research program to an environment that doesn't necessarily require huge amounts of resources. So basically have a research program that's computational and molecular in its nature, and use that to then teach the next generation of smarter scientists to go save the world.
Good plan.
Nice.
I like that.
All right. Before we start talking about your paper, I want to mention our fundraiser, which continues through the end of 2022. This is Spike T-shirts. Celebrate the molecule of the year, the spike protein of SARS-CoV-2, and support science education. When you buy a Spike T-shirt from vaccinated.us, profits go to supporting the science programs of Microbe TV, including this one, This Week in Virology. They have great Spike T-shirts of all colors and for different kinds of people, and they're really cool. Rich had his on last time. We all have one. And you go there, vaccinated.us, put the shirts in the cart, go to checkout, and use the promo code MicrobeTV on checkout, and you will get, that will tell them, you're not going to get anything except a T-shirt. It will tell them to send the profits to Microbe TV. And we're very grateful to vaccinated.us and Matt, who reached out to me originally for doing this. We've got a good amount of money so far. It's really nice. Really appreciate it because we can use support for what we do. That has instantly become my favorite T-shirt. It's got a nice feel to it, right? It's really nice. Yeah, nice feel. And I love the design. It's great. That's really good. I have one as well. So check that out. And by the way, we got John's history. If you want to know Cedric's, you go to TWiV 935. We talked all about his very interesting history. If we did it again, it would use up the whole episode, so we can't do that.
Let's not do this again.
He has a great history.
It was fun. It was a ton of fun.
As John will agree, right? He's got a cool history, right? All right. So tell us, here, the paper is a science paper, Evolution and Antiviral Activity of a Human Protein of Retroviral Origin. So the first author is John Frank, and then we have a number of authors in between, and Cedric is the last author. So maybe, Cedric, you could sort of give us the overview of how this got started. I don't know, was John there at the beginning of this project? Yeah?
Yes. We had actually started a little bit. This was part of actually a grant proposal I submitted to the NIH. It's funded, fortunately. And it was actually, sort of in parallel to all the work we were doing with Nels LD. Of course, you guys know Nels well, at Utah, where we were looking at how, transposable elements, and in particular endogenous retroviruses, regulate innate immunity genes, antiviral genes. And there, of course, here the twist is a different one, and where now we were wondering in this grant proposal, we proposed that some of the protein products encoded by endogenous retroviruses could act as antiviral molecules themselves, because this has been very well documented actually in a number of species not just the envelopes. We're going to talk about the envelopes a lot today. This was the focus of the study, but also, gag proteins that act as that are derived from endogenous retroviruses, then act as antiviral molecules. So that was kind of the idea. But this mechanism, the sort of like we like to think about it as fighting fire with fire, right? This type of interactions have not been described in humans before, and there has been hints that this could be happening, but I would say there was no evidence prior to this work for human gene encoding protein that's derived from retroviruses that has directly antiviral activity against viruses that are circulating now, right? So potentially also zoonotic viruses. In this case, these type D retroviruses that we're going to be talking about. So anyway, so that was like trying to see, can we find something like this in the human genome, since we know that there is this very large pool of retroviral sequences that have been deposited in the human genome in our ancestors, of course, over many millions of years. The potential was there to look for sequences that encode this type of antiviral proteins. So when John joined, I think we had barely started. What we had done, it was only computational at the time. We had started to do a catalog of all these. This is how we started the paper, actually, of all these envelope-related open reading frames we call env-offs in the paper. So we had started like kind of looking at this, kind of skimming through the list using different kind of computational approaches, I would say, to kind of predict which one might actually have antiviral activity of all these envelopes. And so this is when John joined the project. Do you want to take it from here, John?
I can. Yeah. So I guess where I started joining in is basically the idea is can we find a protein that we can actually experimentally test there? And so one of the low-hanging fruits of this project was that there were several envelope open reading frames that have already been annotated as human genes. The syncytins are prime examples of that. And actually, at the time when I joined the lab, a paper had recently been published that described, or maybe a couple of years beforehand, that described a truncated envelope. So basically, it's an envelope an envelope typically mediates entry into a cell, right? So a viral particle is decorated with an envelope and it binds to a receptor and mediates entry. And usually those envelopes are tethered to the membrane by a transmembrane domain. Suppressant actually doesn't have that transmembrane domain. So it's effectively an unanchored envelope protein that we thought still had the capacity to retain its ability to bind it to its target receptor. And so we decided we wanted to experimentally test that one. And it was actually advantageous to us because we already knew what the receptor was for suppressant, because in that original paper, it was reported or suggested that this protein could regulate cell fusion that's mediated by actually one of those syncytins. And so given that we had the receptor, we had the gene of interest, and given that we also knew that there were several endogenous and exogenous retroviruses that are fairly distantly related to each other, but they all share one similarity in that they have an envelope that's able to utilize that same receptor. In this case, it's ASCT2, the neutral amino acid transporter.
So back up for me a minute. Actually, I want to, for just a moment, back way up, because I'm thinking that we have listeners that we acquired during the pandemic and who are unfamiliar with endogenous retroviruses. Is that a possibility?
Sure.
Maybe somebody could very broadly outline what these things are.
Yeah, I could take that one. Try to be brief. Yes, great question. We should have started there. Thank you. So the human genome is a mess. It's a tangled mess. It's stuffed with a lot of stuff, and the sequences that in my lab we really kind of obsess with are these mobile genetic elements. This is a very grab bag kind of terms or transposable elements, which includes endogenous retroviruses. what they all have in common, those sequences, is that they've integrated in the germline of the organism, the germ cells. They must have found their way because they are essentially permanent fixtures in the human genome. Virtually every human being has these sequences, okay? Some, maybe very recently acquired, in which case we may not share them, but 99% of them are shared, okay? So those endogenous retroviruses, initially we know where they come from. They come from retroviruses, those that are infectious, exogenous retroviruses, things like HIV or murine leukemia virus. These are the best known retroviruses, We know that because when you look at their sequence in the human genome, even though they often fragmented, they are remnants, right? They are relics of those. They are, fossils of these ancient viruses. But they look very much like a typical retrovirus, right? So they have these long terminal repeats that flank, the coding sequences, which include, we talked about GAG or capsid proteins, reverse transcriptase, and the envelope, okay? So a subset of those still have all these components. However, they are like, they no longer, so we do not know in the human genome of any currently infectious or even replication competent endogenous retroviruses. So I wanted to bring that point. But in other species, in the mouse or in the chicken, where they were first discovered, those endogenous retroviruses, they are still capable of being infectious and make particles. And in fact, in those same species is where this mechanism that we investigate in the paper, this interference of envelope from endogenous retroviruses against exogenous retroviruses, were already documented in the mouse and in chicken, so way back. So I don't know. I don't know if that answers your question, but basically these are, to summarize, those are the remnants of germline infections of retroviruses. These retroviruses may have been also able to infect other cell types, somatic cells, but somehow, for sure, they were able to infect germ cells or their progenitors in a very early embryo. And I think we're going to return to this point because we think the primary functions of what we found is actually to protect the germline of the organism rather than the somatic cells, possibly also the placenta in this case. So they made their way in, and then they've ceased to be active, at least in the human genome, and they are sort of lay dormant in various stages of decay, basically.
So just one little point is that in the realm of viruses, retroviruses are unusual, if not unique, in that making a DNA copy of the genome and integrating it into the genome of the host is an obligatory part of the life cycle, which leaves open a terrific opportunity for these things ultimately to get stuck, right? So that you're left with a copy, and as you say, they get stuck because, well, they get stuck anyway, okay? But over time, they can lose their activity as viruses. So what percentage of the human genome is ERVs?
Right. So we're talking about 8%. 8% of the human genome. And to put that in perspective, right, it's useful to know that only 1% of the human genome encodes, exons, correspond to the exons of cellular protein coding genes, basically our own genes. And suppressing, so, the question we always have, how much of that the viral genes actually belong to the human gene pile, right? Because, again, we see that they can be co-opted to become now bona fide, human genes. And suppressing was already one co-opted gene that was already annotated. But, the vast majority of what we describe in the first pieces of the paper are not annotated as human genes, right? So anyway, 1% is basically protein coding sequences that encode own cellular proteins, and 8%, so 8 times more DNA, is occupied by these remnants of retroviruses. And you're right, Rich. There are other endogenous viruses that are not related to retroviruses in the human genome. However, extremely few. Maybe you've heard about virus. Was probably one of the first non-retroviral viral sequence described in the human genome that's derived from a completely different group of RNA viruses than retroviruses. But those integration, as as opposed to retroviruses are likely to be very accidental and often the product of DNA repair activities or other activities where they get captured, patched into the genome, sort of in a more random fashion. Yeah, so in the human genome, there is virtually all of the endogenous viruses are retroviruses.
So as you say, it's a mess. I think of it as a bureaucracy, that, it just gets messed up over time. So, John, you said, but I want to return to this. I'm not sure I got it completely. The characterization of suppressin when you first encountered it, was there a function? I mean, the name is consistent with ultimately what you think it does. Is that what they were thinking it does?
So what they originally described it as, it was essentially a suppressor of cell fusion, syncytin-mediated cell fusion. So in essence, it was already kind of serving the function that we thought it would serve as it relates to viruses. So placental cell fusion and placental development requires these cell fusion events, where a single cell will fuse with another cell, creating this multinucleated structure that sort of sits on the outside of the placenta. And that fusion, at least in primates and in humans and several other mammals as well, is mediated by envelopes themselves. So syncytin is actually an ERV-derived envelope as well. And that gene utilizes ACT2 as a receptor. So the idea is a suppressin is blocking cell fusion, or at least regulating cell fusion, to get basically placental fusion to occur in the correct context, by essentially interfering with that interaction between syncytin and the receptor itself.
So was this hypothetical, or did they have real bench experiments to show that it had this activity?
They had some in vitro bench experiments to show that it had this activity. It was a pretty nice paper. They did a good job showing it.
Good.
All right, let me.
But no. Just to clarify, no in vivo evidence. And in fact, I need to mention there is no evidence in vivo in humans that syncytin, we have two genes, syncytin 1 and 2, both of which are derived from different retroviruses. These are full-length envelope proteins that essentially have retained most, if not all, of the activities of a complete active retroviral envelope, which is another thing we can return to, which we think is a very dangerous thing to have. But anyway, they have retained in vitro the activities, right? So they are fusogenic, they bind receptor, much like their ancestral envelopes. But we need to be clear, there is at this point no evidence, in vivo evidence, or genetic evidence that they are involved in placental development in humans. Okay, so no one has ever found a mutation in humans with a placental defect that's caused by a mutation in syncytin. It could well be that it's embryonic lethal. You're never going to see something like this. Yeah. But the evidence is much stronger in the mouse, which has its own syncytins, which come from yet different retroviruses, different endogenous retroviruses, syncytin A and B. And in the mouse, Thierry Heinemann and others have done the experiments there where we can knock out syncytin A and B, and indeed they see placental defects. Okay. So based on what we see in the mouse, based on where the syncytin 1 and 2 in humans are expressed, they express in the cells that John described in this syncytio trophoblast, and based on their in vitro activity to be able to be fusogenic, it's believed that the syncytin 1 and 2 do the same thing as syncytin A and B in the mouse, promote the formation of the syncytio trophoblast. And yet, yes, and indeed, suppressin is yet another player here in that game that appears to be potentially competing with the receptor for syncytin 1, and therefore maybe modulate this fusion, this formation of this syncytium, together with syncytin 1 in this case. And I just want to say also up front, none of those hypotheses, this mechanism is exclusive to what we describe in the paper. Okay, so it may well be what is going on.
Mm-hmm. All right. So, John, you found suppressin, which, as you said, is truncated, so there's no transmembrane, so it could be soluble. The receptor is so the receptor for syncytin is ASCT2, correct?
Yes.
All right, and so you're starting to think that maybe suppressin is involved in that interaction, right?
Yep.
Okay. So you found that. What did you do next?
So the next thing was to basically ask if we express suppressant in some cells, will that confer protection to those cells if they're also expressing ACT2? And so in these initial experiments, we basically took 293 T cells, transfected them with suppressant, and then challenged those cells with basically reporter viruses that we decorated with an envelope from this group of viruses that utilize ACT2. And so the one that we chose was a feline leukemia retrovirus known as RD114. And so basically, when I say decorate, I mean, I think this audience would know we're pseudotyping basically HIV retroviruses that have a GFP reporter in them. And so then we apply the virus to the cells and then just basically counted up the number of GFP positive cells in the presence or absence of suppressant. And basically what we found is that at least in 293 cells, when we have overexpressed suppressant, it conferred protection.
All right. So that's a feline retrovirus that you use there, right?
Yes. There are other members of that group that are exogenous viruses that are actually circulating in old world monkeys as well. But for some strange reason, a lot of these circulating viruses don't seem to cause a severe disease in monkeys. And yeah, that's pretty much the main point with those.
All right. So RD1, you kind of skipped ahead in the paper, but maybe this is actually how it happened, right?
This is how it happened. I should probably go by the paper. I'm sorry.
No worries. I just got confused because
the audience is not looking at the paper, so that's okay.
This was the first experiment to show if suppressing could block, entry from this type of retroviruses. Indeed, and it is right. This is the first thing that John did, and we're very excited to see that it worked.
I mean, makes sense, then right? You want to know you want to do a functional experiment right away. Yeah, makes perfect sense.
So, what are the human retroviruses out there that we are likely to encounter? Right, the HTLV viruses and HIV is about it, right? So this ever served as, I mean, we're probably going to get to this. If this ever served as a functional antiviral thing, it's probably back there in vertebrate history somewhere against some other virus that we're humans at least aren't dealing with right now. Is that fair?
I would say.
You want to go, John? Yeah.
Well, I think we might actually have maybe somewhat differing opinions. Maybe I would say yes, but the thing is we've only discussed suppressant right now. It's entirely possible, and I think this might be where you're going with this, Cedric. Is we don't know what a lot of these other endogenous retroviral proteins encode, and we don't know if maybe some of those could actually be functional against currently circulating viruses. Okay. So it's entirely possible that some of these may be sitting here by virtue of the fact that a lot of viruses tend to like to utilize the same receptors that are very deeply evolutionarily conserved. We might be harboring some sequences that may offer us protection against modern circulating viruses, even the ones that we're currently afflicted by. Is that fair to say, Cedric?
Yes, but I wasn't thinking about that, but that is entirely possible. But I want to go back to the point that Rich is making, that in fact we know remarkably few retroviruses that infect humans that we know of, right? And I just want to emphasize that we know of.
Okay, yeah.
So we don't know what's out there. And the other thing is, like, even though those viruses, like lentiviruses, like related to HIV, if you look for those in primates, try to look for those. Even when you know that some, particularly I'm thinking about chimpanzees, are infected because, you have a strong suspicion that they are infected with these lentiviruses, you can hardly detect them. As you know, lentiviruses also enter latencies. They basically, they might be extremely difficult to detect. So I'm not sure that we have a full understanding of the retroviral diversity that can potentially infect humans.
Okay.
But there is many more retroviruses that infect a whole battery of range of species. And we mentioned, so these type D retroviruses are circulating today in domestic cats, which we are exposed to. They're circulating in a number of monkeys, squirrel monkeys, old world monkeys as well. in bats. Okay, so there are retroviruses out there. Those that make us sick, we know of only not even a handful, but I would just be cautious to not, just, think that this is all the retroviruses that are out there, right?
Yeah.
Okay,
Sure.
I
great.
think they're largely undersampled in all virome studies for technical reasons that we don't need to get into right now. But and they're very just in very low titers often. So they're really actually I think they're largely retroviruses, largely undersampled, underestimated in all viral surveys or virome surveys.
Okay.
Yeah.
So, John, now you had that result with RD114, and forget about the paper. What did you do next?
Well, so the next question was overexpressing this protein only means so much. So we next wanted to know, well, what happens if we actually challenge cells that express suppressant endogenously? And what happens if we remove suppressant? Can the cells become infected? And so we decided to work with placental cells and basically just ask, well, if we knock down suppressant, do the cells become more susceptible to infection with our reporter virus? And that's pretty much what we did. Is we generated a stable knockdown of suppressant and challenged those cells with our reporter virus. And what we found is that those cells became more susceptible to infection with RD114 decorated virus.
Okay.
Can you tell me a little more about those cells? Are they human or mouse cells, and how did
They're
you get
human
them?
jar cells. So the thing to keep in mind about suppressant and actually syncytin as well is we're really talking about genes that are specific to primates.
Okay.
And as regards to suppressant, it's really only specific. It was acquired in the common ancestor of apes and old world monkeys. So there's no real comparison to mouse at this stage with a 1-to-1 ortholog. And so basically what we did is we took placental cell lines and basically first checked to make sure that suppressant was in fact expressed in the cells, made sure that the receptor was expressed in those cells as well. And then only once we confirmed all those sort of underlying basal conditions, then we knocked it down and then tested to see what would happen.
Okay. So that's a good experiment. You knock it down, becomes more susceptible, suggesting your suppression is involved in preventing infection. Okay. Next experiment.
And then if I may add, because to me, the result that really made me happy when John obtained that result, is that then he could rescue it. So he could bring it back. And this is the ultimate experiment because, when you knock down something, you can always say, well, maybe there's all kinds of other off-target effects or other, indirect effects. So what then John did is engineer a transgene to bring back or construct to bring back, to re-express suppressing in those cells that were now depleted partially, and then now restore back some of their resistance. So you kind of move, basically suggesting that the dose of suppressing is directly related to their susceptibility, the cell's susceptibility to these viruses. Yeah.
All right. How about next experiment, John?
Okay. So after rescuing, exactly as Cedric had mentioned, we wanted to do a couple of different things. The first thing since we know that suppressant binds to ACT2 and that there's a subset of retroviruses that all utilize the same receptor, we wanted to see if this protection could be conferred against multiple different or another member of this RDR interference group. And so we basically generated another titer of reporter virus, but this time we decorated it with a different envelope. This one was from squirrel monkey retrovirus, so it's also a member of this interference group, and basically overexpressed suppressant in 293 cells and showed that, again, in the presence of suppressant, you'd protect against a different infection with a different RDR envelope. Beyond that, we also as we were digging through the data and as we were sort of looking for expression of suppressant, we found that it was also expressed earlier on in development. So we found that there were some transcripts and we found expression of suppressant in human embryonic stem cells. So we also just wanted to ask, well, could human embryonic stem cells be susceptible or resistant to infection? And we found there, at least in our hands, that at least if you challenge those cells with RD114 decorated virus, they also tend to be pretty resistant to infection because we think that they're expressing suppressant. But we weren't able to do a lot of the sort of experimental manipulations that we would like to do with the placental cells in that case. H1 embryonic stem cells are just so hard to sort of manipulate and sort of genetically modify at that point.
Okay. So I think you also did a lot of expression experiments, right, to see where this protein is being made. Can you tell us about those?
Yeah, so this was actually done in collaboration with a lot of other people who really helped make this possible. So first things first, we wanted to understand. So we just happened to see this just in a data set on the genome browser, and so we just wanted to explore what was going on here. And so we mined all sorts of single cell and bulk transcriptome data sets, as well as various different genomics data sets, ChIP-seq, DNA, ATAC-seq, DNA successibility, all those different types of omics experiments. And basically what we did is we wanted to see where is suppressant expressed and at what stages of development. And we'll start from early on to later. What we found is that suppressant is expressed at early stages in the pre-implantation embryo. And that expression persists through embryonic development to implantation. Obviously, we can't show this physiologically. This is basically just what we're seeing at the different stages of embryonic development, and then throughout placental development as well. And so what we find is that its expression seems to be driven by its own LTR. In addition to that, we also find that its expression, particularly in the placenta, starts to differentiate from, say, syncytin 1. And so basically what we can see is just this really beautiful pattern of suppressant persisting throughout early development. And so that suggested to us that perhaps suppressant could also be protecting us all the way to those early stages of embryonic development from viral infection or germline invasion. Please tell me if there's something that was unclear there.
No, so later, you don't see it later in development, basically, right?
You tend to. We didn't look at fetal tissues beyond that early pre-implantation embryo stage. If you look at various different tissues, if I remember correctly, you will see some suppressant expression, very low levels, I think in the testes. And I'd have to go back over the data. I think testes was the main one, and I think there might have been a little bit in immune cells, but I'd have to double check that.
Yeah.
And then
not
beyond
much else. Yeah.
Beyond that, we also looked for.
Okay, sorry.
Go.
I wasn't sure you were going to mention this, but to me, it's like one interesting follow-up. In fact, I've been thinking about this all week. All week, I've been thinking about this because we had a visitor here giving a talk on metabolism, and he saw our story and he got really excited for very different reasons. Let me tell you just in a nutshell, since we're taking a little detour.
Yeah, sure.
It turns out, so suppressing binds this receptor ASCT2. We did not show this. This was shown by a Japanese group prior to our study, but all of our results are consistent with this protein about to bind that receptor ASCT2. ASCT2 does, of course, has a cellular function. It's not just here to be bound by viruses or whatever. It is a neutral amino acid transporter. It is actually the major transporter of neutral amino acid like glutamate into the cell. And it's a housekeeping gene, by the way. The receptor ASCT2 is broadly expressed, not just expressed in the placenta. And it's basically what I learned this week. I didn't quite appreciate how important it is in metabolism. It's basically a switch in metabolism that that regulates glucose metabolism, homeostasis. If you shut off that path, you would favor glucose. I'm trying to remember what I just learned today or yesterday. And so you would shut down, basically it's a major metabolic switch. Okay. So now this is when I think it becomes interesting because it is there's a pretty good I'm speculating right here. There's a good possibility that ASCT suppressing would affect the function of the receptor. And the reason why, and we haven't tested this, but this is really what I would like to look at now, because it's been seen that the retroviral envelopes that we mentioned, RD114 from the cat or the one from the monkeys, experiments have shown that these envelopes, when they bind that receptor, they do actually perturb the function of the receptor. It's not like they block it, but it doesn't function as well. It doesn't import amino acid. So it's a good reason to think that, reason to believe that it's possible that ASCT2 is also has a physiological function here, completely independent from like being an antiviral or an anti-syncytin, in switching metabolism. And now back to the expression. One piece of data that's not in the paper, but I would offer it to the listeners today, is that this suppressin is highly overexpressed in multiple cancers. Turns out, in particular, if you remember, John, it's really, really overexpressed in testicular cancers, tumors, like massively so. So I'm really, really curious at this point about what is the effect on metabolism in those cancers when suppressin is really just abundantly expressed. One would predict that if it has an impact on the function of that receptor, it may have a major impact on metabolism, which is very important for tumorigenesis and metastasis.
So that's something you could look at easily.
Yeah, exactly. I think it's really testable.
Yeah.
And that would be really interesting to look at. So I just wanted to offer that additional unpublished observations.
Yeah. But the idea for suppressin is that it's binding the receptor ASCT2, and that's just physically blocking virus infection, right? That's straightforward, right? Yeah.
Yeah.
But in light of this, remind me, maybe you already said this, but what's the overall expression profile, both during development and tissue-wise of suppressin? Because if it has any function as a metabolism regulator, I would expect that to show up in its tissue distribution and its developmental distribution. So what can you say about that?
During early development, its expression emerges at about the morula stage, so pre-implantation embryo, and then it persists through the development of the trovectoderm. So that's the tissue type that will give rise to the placenta. And during that stage, it'll persist throughout placental development. And as the tissues start to differentiate, it'll become more present in the extravillous trophoblast. And if I remember, extravillous trophoblast specifically. As far as in adult tissues, if I remember correctly, testes is where you will see its expression to an extent. And then pretty much what Cedric had already mentioned as far as its expression in cancers is concerned. That's what I can remember so far. Yeah, I think that's about it.
So it's possible that it is regulating metabolism during placental development. And I know nothing about metabolism in the placenta. I don't know if it's different than other tissues. I don't know if you guys know anything about this, let us know. But I'm going to dig. This is a very new idea, okay? So this is something I'm really curious to dig into the literature here. For example, we know that suppressin is most highly expressed in the most invasive cells of the placenta, the extravilli trophoblasts, they are called. If we look at the single cell level, through single-cell RNA-seq. I'm talking about RNA levels. Proteins, we don't have that resolution, actually. Even though Caroline Coyne, one of our collaborators, who is now at Duke, provided some beautiful protein staining of actual human placenta that shows that suppressin is broadly expressed, but we can't access this extravilli trophoblast. They can be obtained at delivery, essentially. So they are the ones that are the interface with the uterine wall of the mother, and those are very invasive cells. So it is possible, and in fact, and it's actually a form of. It's sometimes in the literature on placenta. This is compared to metastasis, actually, because this is an very aggressive invasion of a tissue, and so it's possible that there is metabolic switches going on. I'm really speculating here because that's the fun of TWiV, right?
Yeah, sure, of course.
It's possible that there is something special about the metabolism of those cells, and maybe ASCT2 to shut off ASCT2 essentially might be important for that process of migration. Anyway, all of this is very wild hypotheses, but they are testable. I think that's the cool thing about science, right?
Sure. Okay. Let's see. What do I want to ask you? So
we moved away from viruses, huh?
That's okay. It's not a problem. So there's a part of the paper where you talk about when this might have happened, right? The insertion of this suppressin gene by looking at genomics, right, John? Can you talk a little bit about that?
So basically, the question there was, we wanted to ask, well, when was suppressant acquired?
Right.
And this is really a proxy for giving us some sort of idea as to how physiologically relevant or how biologically relevant is suppressant. Is this actually a domesticated gene? And so the way we go about that is we basically ask for the location of suppressant, is it in the same location across closely related species or across other species? So is it an ortholog, basically? And so basically what we found is when we looked across different primates, we found that the suppressant encoding gene and sort of the endogenous retrovirus that encoded for suppressant, or the ancestral envelope of that gave rise to suppressant, that was present and conserved across apes and old world monkeys, but was notably absent in new world monkeys, which basically told us that our common ancestor of apes and old world monkeys is a species that originally acquired the retrovirus that would ultimately give rise to suppressant. So that's the first thing. The second question is okay, when did suppressant emerge? How did suppressant emerge in the first place? And so to look at that, we basically looked at the length of the open reading frame and asked, where did the stop codon emerge that ultimately truncated the envelope gene into suppressant? And there, again, we see that the stop codon sort of emerged in the common ancestor of apes and old world monkeys.
So that's
following. Go ahead.
How long ago are we talking then?
15 to 20 million years, I think that's about right.
Okay.
Yeah. So then the other thing we wanted to ask, well, okay, so you might have this gene that's present in an ortholog, but how do you know whether this gene has actually been preserved by natural selection? How do you know that it has a function? And so basically what we can look at is looking at the preservation of codons across the entire length of the gene and across species. And what we find is that actually in apes, the gene has been very, very nicely conserved and sort of been preserved, as you'd expect. But in old world monkeys, you start seeing a lot of variation, not only in the length of the gene, but also in the identities of the codons that are present there. And so that led us to sort of conclude that really suppressant has really been preserved by natural selection in apes over extended periods of time, speaking to the idea that this probably has not only an antiviral function, but may also have some other function as well. And we've already speculated about this quite a bit during this conversation.
Right.
So is that evolution the same or similar as the evolution of the cognate syncytians and the receptor?
I'll start off with the receptor. The receptor is very, very deeply conserved. That thing is an ancient gene that's preserved across vertebrates, I want to say.
Okay.
As for the syncytin that it sort of has been proposed to co-regulate, that gene was actually acquired in the common ancestor of apes. So the evolution of suppressant really nicely sort of matches what we see for syncytin 1.
Okay.
Not exactly. No. You remember, like, it was deleted from the old world monkeys.
Oh yeah, sorry. My apologies. It was acquired in the.
That's actually it's a detail, but it's actually interesting because it turns out so this is interesting because we cannot tell at the moment which one came first, suppressing or syncytin, and this is to us very important. I'm going to try to explain why. I'm going to get into evolutionary points here. So since
we're talking about two different retroviral acquisitions, but that are related. Okay,
that's right.
So which came first?
Yeah.
This is cool. I can see why this is interesting. Okay.
Because the one way to think about the story that we depict about suppressing. So there's a paradox here. The paradox is what John mentioned is like, for an antiviral protein, you would expect that maybe, selection will be relatively transient. You will protect, like you said, back 30 million years ago or 20 million years ago, and then virus is gone. There's no need to keep suppressing, right? Why would you keep suppressing? So yeah, it's possible that then there is a co-option to a physiological or developmental function and you maintain this. That's a possibility. We discussed this. Another one is that once you've acquired syncytin, potentially you've got a problem here. And by the way, I need to mention there is a beautiful perspective article accompanying our paper. I don't know if you checked that out Ricky Padilla and Rick McLaughlin.
Yeah.
It's a beautiful piece because they in fact go at length in discussing this relationship and this issue, this potential problem that having syncytin poses, because now you have, as I mentioned earlier, a fully functional envelope protein that's expressed in the developing placenta that could package all kinds of things, including endogenous viruses, exogenous viruses.
Okay.
By the way, syncytin has been shown experimentally to be able to, you can pseudotype HIV with syncytin. Okay, so it highlights the challenges that you face here. So we like to think that having suppressin was essential to, in fact, the co-option of syncytin to begin with. And it's even possible that it preceded the invention of syncytin and it was required.
Yeah.
If so, then the prediction would be that in species that, other species that have syncytins, such as the mouse, such as the dog, independently co-opted, that appears to do all the same thing, they should be also regulator of this process. Not only, you can package, but you also start infusing all kinds of cells. I mean, it's a mess too, right? So this is why we really would like to know which one came first, because in the prediction under this model that I described would be that perhaps then suppressin came up first. Maybe it had an antiviral function to begin with, and that was beneficial against its own kind, right? The retroviruses that it derived from, and indeed, the extension of these viruses, at least in the germline, coincide with the emergence of suppressin, right? So it's conceivable that suppressin defeated the virus that it came from, but then enable the co-option of syncytin now from yet a different type of retrovirus to do this essential function in the placenta. Of course, this is going to be very hard to test this, and I'm afraid it might remain speculative, but that's an interesting way to think about it.
Yeah.
Why was
check out the Perspective article. There's a very nice. It's very nicely explained, better than I tried to do it here.
So why was suppressin lost in owl monkey lineages? Do we understand?
So can I take that just really because that's the beauty. It's like in fact, both suppressin and syncytin were derived from retroviruses endogenized in the common ancestor of hominoid, the apes, and old world monkeys.
Okay.
But both of them have been lost after this in all the old world monkeys. Syncytin was lost because there's a deletion in the genome of those species that removed physically the gene. The gene is gone. And suppressin, as we show in the paper, is essentially a pseudogene in old world monkeys that has experienced, relaxed selection. So there's really a strong coincidence and co-evolution between syncytin and suppressin.
So if you lose suppressin, you don't need. Wait, sorry. Which one went first? We don't know.
If you lose syncytin, you don't need suppressin.
You don't need suppressin anymore. Okay.
Yeah.
So how do they make placentas then?
They have another one. They may have syncytin too.
Okay.
So they have another one. They may have different placentas. So the beauty is of the story of the syncytin is like the reason why rodents have two, and they are only partially redundant. They take care of different layers of the placenta. Essentially, this is my understanding. And so it's possible then so first of all, it's possible that the old world monkeys have yet acquired another syncytin. So it seems to be like a revolving door of these syncytins, right? And no one understands why is that, right? Because the question is also, how was the ancestral mammal regulating this process? Presumably also at a placenta and also maybe at a syncytial trophoblast. Yet we don't know if an ancestral conserved syncytin across all mammals. They all are lineage specific, just like they would have evolved over and over. So it strongly suggests that there is a turnover, sort of a revolving door of these proteins. So it's possible that the old monkeys have an additional syncytin-free, if you want, that we just don't know about.
Yeah.
Or they have a placenta that's slightly different because the placenta is also the most rapidly evolving organ of all of mammals. Okay, it's a fascinating organ in that way. It's extremely variable anatomically. Some are very shallow invasion. Some, like the human placenta, is a very deeply invasive placenta, quite different actually from the old world monkey placenta, which does not invade the maternal tissue as deeply. So you can see why there's multiple reasons for why they would have ditched that order. That's syncytin.
Got it.
Yeah.
We don't understand the reason for these anatomical differences in the placentas, right?
I do not. I don't know.
Okay. Must be some reason why one is deeper, shallow
Yeah.
versus the other, right? There has to be a reason, but yeah, we don't know. All right. So the other thing I wanted to ask you is that you make this statement towards the end that most in humans, most of the endogenous retroviruses are now moving around by retrotransposition. They're not infecting us exogenously, right? So you say we predict the adaptive benefits of these ORFs like suppressin would be transient, right? When the virus first comes in, it may help, but then for viruses that are already endogenized, it could be of no use whatsoever, right?
Yes, I would say that that's probably a fair statement. And there is some evidence to suggest that. So there's a paper out of Paul Bianach's lab that came out quite a few years ago that looked at HERV-T. And they actually showed exactly that kind of thing where they basically showed that ancestrally this HERV-T existed and that it likely provided this protection. But then with that loss, you kind of lost the functionality of that HERV-T over time.
So are you saying that suppressin might be protecting us against unknown RD1, maybe RD114 related retroviruses that may impinge upon us periodically? Maybe they don't get anywhere because of suppressin? Is that an idea?
I hesitate to say that that's the only function that it has, right?
Okay.
I think, yes, absolutely. I think it can protect the germline and protect the developing fetus from infection. But as we've already sort of discussed, it's intertwined with sort of physiological functions that we have here as well. And that's why it's been preserved over time. So I would say, yes, it can protect, but I'd say yes, and it probably has other functions, which is why it's still present in our genomes and has been preserved by natural selection.
Are there suppressin-like functions in the mouse so that you can knock that out and ask what impact there is on placental development? I'm interested in what evidence that these actually regulate the fusion function and what happens if you knock it out.
I don't know of a study that has reported that. I know that the Japanese group that originally described suppressin was looking into that at one point, and they might have found one, but I've never seen the paper finally come out. I think that's probably a field still for active study. Please feel free to jump in, Cedric, if you know of any other studies, but I haven't heard of anything yet so far. I think that's sort of an open question.
Yeah. You mean a functional homologue of suppressin, right?
Yeah.
Yeah. No, that would be that's what I tried to explain earlier, that there could be that would be a prediction that species that have this mode of syncytial trophoblast, development through this syncytin-like activities, they would need some regulators as well. And it might be very hard to find because, this, so we can go back now to the early part of the papers where we explore that sort of like pool of envelope sequences encoded in the human genome, right? So we can kind of return to this bigger picture here. What else is in there, right? We have about 1,500 candidate envelope sequence that may encode proteins that can bind receptor at the sort of like this would be our prediction. They have the surface domain, right? That was sort of our filter. We scheme the genome and ask, what are the open reading frame that correspond to the N-terminal region of an envelope, which is where the surface, the receptor binding domain is. And we came up with 1,500 open reading frames. We've shown then that about half of those you can see clear evidence of transcription somewhere, of transcript, often very tightly regulated, which is no surprise, at least to us, because all of these endogenous retroviruses are not randomly expressed. They're extremely tightly regulated during development and in immunity. That's something we've looked at in the past. But how many of those actually encode proteins? We do not know that. We would love to know that. And how many have physiological or antiviral functions? We do not know that. But I think that's the next step, is to go now systematically, for example, through a genetic screen, look for the functions or the biochemical activities of those proteins. And I want to emphasize that they don't need to be genes that are important to human physiology to be interesting molecules. To me, it's fascinating to think that our genome might encode a reservoir of antiviral proteins, which have, potentially interesting properties, not on just properties of antiviral, but other like physiological properties that just we don't know anything about, right? So I like to think about that just huge pool of sequence that we have. And what about GAG? What about the other products, right? There's just as many of those as well that just no one has studied.
So along the same line, you state in your introduction that most NVORFs are expressed early in human development, and that with the exception of the brain, they're rarely expressed in differentiated tissues under normal conditions. So there's a burst of transcriptional activity of these things very early during development. What do you make of that?
I think that, you could think of it this way, is that these things may predominantly be protecting the germline initially, right? That initial burst. Obviously, a lot of this could just be transcriptional noise because we know that the genome is activated and you get a lot of these ERVs being expressed. But the germline, and especially that early embryo, is a very sort of vulnerable organism and vulnerable tissue. So by having this initial expression before you have all of your immune systems set up, I think that could be sort of a way of providing a little bit of initial protection.
So that kind of gets at the question I was going to ask, which builds on your last sentence, which I want to read, except it doesn't start with, "It has not escaped our notice that," but that famous preface. But the human genome may hold many other retrovirus-derived proteins with protective effects against viral infection. Yeah. And so I wanted to ask a little more about, do you think that we're going to find that these are useful against viral infections at other times in development, later in life?
It's a great question. I could envision, by virtue that we do see quite a bit of expression in the testes and sort of the germline there, I could envision those being sort of local sites where we have really, really an ideal place to have protection by these endogenous retroviral sequences. Beyond that, the physiology gets really complicated. And so I'm not quite sure. I think there's a lot that you have to sort of consider in that way. Maybe individual cell types might be protected. Say, for example, retroviruses that predominantly infect immune cells. If you have expression of some of these ERV envelopes in those immune cells, that might actually provide some degree of protection. So I think it's definitely an open field of study, and I think I can envision ways in which they can be protective at different stages.
Yeah. Just to add to this, there is a piece we didn't talk about that we document in the paper, is that a number of those envelope transcript are upregulated by immune challenges or activation. Right. We have a whole figure of how in HIV-infected cells, you have a subset of those that turn on. And this was, again, not a big surprise because we've seen, us and others, that endogenous retroviruses are inducible by infection. And there is an extensive crosstalk between exogenous and endogenous retroviruses that is a regulatory crosstalk that, again, us and others have documented. So it's conceivable that, in some viral challenges, some of those envelope might be protective. Yep.
And there was also that recent Coffin paper that came out that also sort of describes some of the tissue-specific expression of some of these ERV envelopes, as well as other HERV-K GAGs. So there's definitely some expression of these envelopes in somatic tissues as well. I just think it's going to be an open field of study.
So basically, you're thinking that if an immune cell is recognizing a pathogen, then this is among the genes that are turned on because it may have protective activities, right?
Exactly. It could almost function as sort of like an innate restriction factor or something like that.
Okay. Now, Cedric, are you planning to look at some of these other envelopes and try and figure out what they're doing?
Yeah, we would love to. Yes, I'm just waiting for another John. These are hard to come by.
Understood. Yeah, you can have all the great ideas in the world, but if it can't execute
somebody's
them,
got to do it. That's right.
Somebody has to do it.
or I'll take a sabbatical maybe and then just get at it.
Do it yourself, yeah.
No, you probably don't want that.
Depends. Is there anything we missed that we should talk about?
No, I think we've covered
I
a
think
lot.
we covered
Covered it
John?
all? It's good. Is it good analysis? All right. I want to make sure we did a fair and comprehensive analysis. All right. Well, Cedric Feschotte, Cornell University, thank you so much for coming back. Really appreciate it.
Thank you for having us.
Yeah, and when you have another cool story, let us know. I know you have some cool stories you're working on. You told me about them, and one of them involves zebrafish. That's pretty cool. So keep us in mind, okay?
Endogenous retroviruses again, but this time in fish.
Yeah.
So that's not a whole other story.
That's
coming
cool.
up.
I like that story. And John Frank, Yale University, thanks so much, and good luck there in your postdoc.
Yeah.
Thank you for the invitation.
Yep, pleasure.
Thanks.
Fascinating stuff.
Cool stuff.
What's
Really
amazing
cool
is how all these databases of expression, you just tap into it. They're public databases, right? Yeah. It's such a resource. But as you said, you have to get people who know how to do it.
Yeah.
You can't just let's access this CHIP database.
And it's huge. So for somebody like John, who's interested in teaching, you can be a computational biologist.
Yeah.
Okay. And it's a low biohazard, low cost, I would think, relatively.
Yeah.
Thing that you could do without a lot of, capital equipment and stuff. Great.
And some of the experiments are straightforward, making pseudotypes and infecting cells with a reporter. It's not dangerous, right? You could do that with an undergraduate-driven, research team. But also, the computational part is very accessible, yeah. Well, not to me, but it could be to others. I would have no idea. I left active research before those computational methods got really going. Let's do some email. Now, the first one here, unfortunately, when Dixon came back from his power outage, he wasn't recorded because, he wasn't recorded. So everything that he said was gone. So I had to edit it severely as a consequence, because there were many places where we were responding to him and all that had to go. It was still an hour and 40 minutes, but anyway, this first email was read by him, so nobody heard it. So let me read it. It's from Brenda. Have been listening to your shows for years now. Always appreciated. Parasitism is my favorite. Such bizarre worlds within worlds. My resistance to getting vaccinated started this past year, mid-January, when a sloppy booster shot with a too long needle caused immediate and ongoing problems. I'm 71 and in great shape, 25 plus years into teaching karate, and for the last few years have been doing 20 to 30 hours a week of carpentry work. I'm as stoic as they come. The mid-January 2022 shot gave me a 24-hour headache to die for. I don't ever want to go through that again. Every cell in my head hurt, the roots of my eyelashes and every other hair, every tooth like it was being pulled. My earlobes were screaming. That's very descriptive, isn't it?
Yeah. Reminds me of a Shelley Berman routine. My tongue is asleep and my teeth itch. He was describing a hangover.
For several months, my neck, shoulder, and arm were in enough pain to disrupt sleep, nor could I get my physician. I had one then. She's quit, so like 1/3 of people in my province, that luxury is gone, to see me in a timely manner. Not until July were the ultrasound, x-ray, and physio lined up. The physio has helped, plus working on range of motion and letting time work its magic. I think that's really a good one, letting time work its magic. Oh, everybody remember that when you have issues, sometimes you have to let time work its magic. Pain is gone now, but the neuropathy continues, tingling down my arm to fingertips, plus the arm and hand go numb when I get into a deep sleep, which strangely ache as I awaken. This can cause permanent damage, but I'm hopeful it will slowly abate. So, will I get another shot of any kind ever? Nah, don't think so. The odds may be 1 in high odds that the next needle does physical harm, but hey, I don't want to have to watch the expert vaccinating like a hawk while telling them how to do their job, and B, risking in the least having anyone hurt me again in that way. While the mechanics of vaccinating isn't exactly your bailiwick, perhaps you could do a bit on how it should be done to avoid injuries and what the literature says about the incidents and consequences of mindless jabbing. Cheers for your fascinating work, Brenda, who is from Canada. So, and I did say last time, this is not our bailiwick, and I don't know anything about it. Daniel has talked about proper needle techniques in the past, and I don't know anything about the incidents and consequences of mindless jabbing. I know mindless yabbing, when people talk too much, when not jabbing. So, and I
remember that. Go ahead.
I don't think either of you know anything about this either, right?
No, but I do.
I'm sorry, Kathy, go ahead.
I remember that Daniel talked about, especially this was coming up about worry about whether the needle was hitting a blood vessel. And were you taught to pull out the plunger or whatever it was, and that there was a debate even among the physicians about how they were taught and how it should be done. And so, like Brenda says, either, watching them like a hawk and trying to tell them how to do it. But yeah.
Well, go ahead.
I know little enough so that I question how much of this has to do with the vaccination technique and how much has to do with the vaccine itself. Because, as I recall, this is Moderna, right?
Yeah. Well, she didn't say, actually.
She didn't say. Okay. At any rate, people have nasty reactions to some of these vaccines sometimes, okay, that can linger. So I'd like to know, you haven't asked Daniel about this, right? Whether there is potentially, genuinely a relationship between the vaccination technique itself and this sort of reaction. I don't know.
Well, Daniel didn't hear this last time because it was cut out, but he will hear it now. So if you have any, Daniel usually responds in the next clinical update, so that'll be next week. So you could say something. But, when to a certain extent, to a large extent, you put your hands in trust of someone else, right?
Yeah.
Every time I go for a vaccine, I'm like, okay, do it, and it could screw up or not, but I'm putting my trust in you. That's how the system works. Same thing when you get on an airplane. You put your trust in the pilots, right?
Same thing when you walk on the street. You're putting your trust that cars aren't going to run off the road and run into you.
Yeah.
Yeah.
All of that is the way life works. And if you don't do that, then life can be very difficult, right? If you don't trust anyone. So I've had no problems with vaccinations, but who knows what will happen. So I appreciate your concern, Brenda. All right. Kathy, can you take the next one?
Larkin writes, JFK plane wastewater testing with a relevant quote. To supplement swabs from travelers, this summer, Concentric began taking about 1 liter of wastewater from each long-haul international flight landing at JFK for testing as well. Wastewater surveillance has become a valuable tool in helping cities in the U.S. keep track of COVID-19 cases, as well as variants, and could also be a critical part of monitoring incoming pathogens on flights. And it says this Time article quote is from some archive and gives the link. So you can check that out. I haven't.
Yeah, it's an archive of Time magazine. Yeah, cool. I think this is really a good idea, right?
I really like that. Yeah.
Mm-hmm.
I didn't think of that. You take a little of the stuff in the plane and you look, see what's there. And then what do you do, though? That's the thing. What do you do? If you find some weird...
Publish.
If you find Nipah virus, right? What are you going to do? Because
obviously you can keep track of where the plane came from, but you have no idea about the hundreds of people on the plane, where they were before that flight.
And where they're going. You don't know where they're going for the most part.
Yeah.
But I can imagine you would find all sorts of things, and it may not be relevant, right? So although it's a valuable tool, I think it has to be used wisely.
Yeah.
Right. Rich, can you take the next one?
Rich writes, I know the TWiV team is interested in education, so I'm passing this along. A friend who lives near me shared this note. Later, the story broke in a variety of publications. Quote, one of my biotech students is famous. He's the first person to sequence the freshwater angelfish genome using tissue from his pet fish when it naturally expired. And he gives a link to a New Scientist article that describes this. Here's a link to the paper on his research. He was able to do this using the resources of BioCurious, a community research lab. There he used an Oxford nanopore to generate a long-read sequence of the fish genome. Holy cow. Many other resources are available there. This is what can enable the next generation of researchers to do work even when in high school. We had a letter from a high school student back earlier about where to go next. Oh, this is amazing.
Yeah. Isn't that something?
Yeah.
So I'm interested in this community resource. BioCurious, Santa Clara, California. And basis, independent Silicon Valley. Let's look up BioCurious. BioCurious.org. Oh, no information.
It's a community space for biology. I don't know where they are located.
Welcome to the world's largest community lab space for biology in the heart of Silicon Valley.
Yeah.
Holy cow, this is wonderful.
Yeah, there are many places like this. There's some here in New York as well. Yeah.
You know, I suppose, well, this is the first time I've heard you can use a nanopore apparatus and get long-read sequence on your dead pet fish. That's awesome.
Yes. I didn't know you could do things like that. I knew this is
amazing.
You could make plasmids and put them in E. coli and so forth, but that's great. So I think this is good for schools, right? Where schools don't have a lot of resources, but you could go here and do this. That's great. Thank you, Rich. Very cool. Kevin writes, on TWiV 954, which was last Friday's, there was this email with a link to a paper entitled Serious Adverse Events of Special Interest Following mRNA COVID-19 Vaccination in Randomized Trials in Adults. So this is the paper that, Rich, you read the email, and we weren't sure what to make of it.
Right.
Dr. Susan Oliver and Dr. David Gorski did a really good job debunking that paper, and he includes links to both. Good. So thank you very much. That saves us from having to do it. So go check those out.
Yeah, we were sort of doing that on the fly. And what's this? He's got another link here.
Anti-vaxxers fooled by it's a video, by hacking and apples to oranges comparisons. I don't know what that means.
Well, just from the title, it sounds like it's talking about how these anti-vaxxers who wrote the paper were, I don't know.
Yeah, so this is the preprint claimed to show vaccines cause more harm than good, but it doesn't show that at all. In fact, it doesn't really show anything at all in this video. Susan Oliver, accompanied by Cindy the dog, goes over it. Okay, so that's good.
Hacking. I'm sorry.
You know, that's like when you do the statistics and you're trying to get to p-value of less than 0.05, and that you keep doing things so that you can get to the p-value that you want.
Misuse of data analysis to find patterns in data that can be presented as statistically significant when in fact there is no real underlying effect. All right. Well, thank you, Kevin.
Yes.
That's very useful.
Kathy, can you take the next one?
Laura writes, hi, TWiV team. I've been listening to your show for about a year now. It is so helpful to be able to listen to experts who are earnestly seeking answers to the important questions. I'm a chemist at the Pennsylvania Department of Health and a virology layperson, but I've had the privilege of helping out the virology team during COVID surges by prepping PCR samples. I also run a folk dance series in Delaware, which is a high-risk activity for COVID transmission, and we're doing our best to prevent transmission, especially because our ages run from 8 to 90, and our community is extremely cautious. I'm curious if people can monitor for asymptomatic infection in the following way. If a person chooses not to get any further vaccinations, is it possible for them to pay for antibody testing on some regular basis and look for increases? Does that confirm if they have had a COVID exposure? If they have had an asymptomatic COVID exposure, what does this imply about their need for worry on exposure to future COVID, barring significantly more lethal or infectious variants? Thank you so much. I'm referencing you a lot in my conversations with our other COVID-curious folks. Cheers, Laura. So the part about whether a person chooses or not to get any further vaccinations, I think is, I'm not sure where that fits in, but is it possible to pay for antibody testing on some regular basis and look for increases? So at this point, I don't think there's any way of getting testing for antibodies, and you'd have to get testing specifically for N or some other SARS-CoV-2 antigen, not the spike, because spike is what's in the vaccine. So can you pay for antibody testing to look to see if you get an increase, which might imply that you had gotten infected? So I think we also don't know enough about antibody levels. I think we don't know enough about antibody levels for spike, let alone for N, for which there's probably a lot less testing, to be able to convincingly say whether someone had a COVID exposure or not.
So
go ahead.
Go ahead. We discussed this or something like this recently because I was boasting about not having been apparently had COVID.
Yeah.
At least a symptomatic infection. And I do get bled occasionally because I'm part of a study. Okay. And they test for N and S. And I am so far N negative. And that raised the question, could I have had a glancing blow and be N antibody negative? We didn't know the answer to that. And then Daniels, of course, sent us a bunch of references that says, a fairly high percentage of people who've had PCR positive but asymptomatic infections are N antibody negative. Okay. Even if you could, and I don't know whether you can just, go to your local clinical lab and say, I want to be tested. Okay. I kind of doubt it. And it probably wouldn't be cheap. But even if you could, I'm not sure that that's going to be a real good diagnostic for whether or not you've had an exposure.
Yeah.
In particular, an asymptomatic infection.
Yeah, I think you'd have to test every month at least to do that, and that's not really practical, right?
So what were we talking about a little while ago about you put your trust in other people? We are entering, at least in my experience, in my environment, in my life, okay, we're entering the realm of, pretend it's normal and cross your fingers, right? We just dropped the vaccination requirement for attending our chorus rehearsals.
Mm-hmm.
There you go.
Who's next? Rich, can you take the next
Lisa
one?
writes, thanks for the wonderful podcast. The recent discussions of monoclonal antibody drugs losing effectiveness for COVID got me wondering whether there are polyclonal antibody drugs for viruses. Is that approach used, or is there a reason monoclonal is better than polyclonal? It seems intuitively like it would be harder for a virus to beat a polyclonal drug than a monoclonal drug. Lisa, who is in Austin, Texas. I'll be darned. Lisa, let's do lunch. I'm always looking for an excuse. So, first of all, polyclonal, I can think of two kinds of polyclonal. One is convalescent serum, at least two kinds. Convalescent serum from somebody who's been infected, okay?
Yep.
And so that would be a massively polyclonal and fairly uncharacterized serum. So you don't really know what you're getting other than it's somebody that recovered from an infection. And there were all kinds of studies early on in the pandemic about whether convalescent serum was effective. And basically, I think the bottom line was, meh, not really necessarily. Okay.
Yeah.
However, then monoclonals come along. So monoclonal is basically you can, by a variety of techniques, make preparations that contain antibodies that are of a unique type, that have a unique specificity for a unique protein. And quite often, so a monoclonal, a true monoclonal drug would be a preparation that contains just one of these.
Mm-hmm.
However, a lot of these preparations contain mixtures of different monoclonals. So they are kind of polyclonal in that regard.
Yeah.
So
Evusheld
is
is
an
too,
approach.
right? Evusheld is too. Unfortunately, some of the Omicron variants are resistant, so to both
of
so
them.
it doesn't work. But if I remember, we had Arturo Casadevall on, right? He was talking about convalescent serum. It never really took off, did it?
Right.
It was used early on when we didn't have anything, but it seems to me that it wasn't so good.
Yeah, there was a lot of debate about it. There were a lot of people who were really enthusiastic about it. There were some studies that suggested it might be useful, others that didn't. It's one of these typical things where, maybe not. And then, other therapies sort of overtook the whole thing and were obviously better.
Let me read one more from Philip. You have been my link to scientific information since the beginning of the pandemic. Thank you so much for making your expertise available to a worldwide audience. I watched with great interest TWiV 950 with Richard Knight regarding prion diseases. Just a note that restrictions in Japan, which used to preclude a person who had only spent one day in the UK between 1980 and 1996 from giving blood, have been relaxed. It is now 1 month or more for the UK. Restrictions for other countries and times are still enforced. Interesting. So if you've lived in the UK a month between those years, you can't give blood in Japan. That's because you could have a prion disease derived from BSE. An interesting response in Japan to the BSE outbreak was that a comprehensive tracking and tracing system for all cattle has been implemented, which records the exact lineage, birthplace, date of birth, etc., of every cow, steer, and bull in Japan. At many supermarkets and restaurants, they will list the registration numbers of the animals on display. At fancy restaurants, they might even have the registration papers on display to verify the provenance of your steak. Regional beef is a big deal here. In any case, I thought this was an interesting response to prion disease. Philip, who is over in Kyoto. Oh, that's very interesting. Only in Japan would they record every cow, steer, and bull, right? Because here in the U.S., we have no idea.
No. And I'm thinking, I haven't heard anything about BSE for a long time. I wonder if there's still at all any kind of a problem. Do you know?
No, according to Knight, that there's haven't been any more detected recently, no. But he also said, it depends on your surveillance, and he wasn't convinced that the surveillance was the best. But he thinks that outbreak is over. But whether you have cows developing spontaneous BSE and they get into the food chain, he doesn't know. Could happen. It's very interesting. All right.
Two things. Two reasons why I've gone to my Red Cross donor app.
Yeah.
But it's just not responding. But I know, or I'm virtually certain, that the amount of time that the U.S. Says for having lived or been in the UK is 90 days. And then, because I calculated once how long I'd been there because of all the DNA tumor virus meetings that were in Cambridge, and it was less than 90 days. So I was okay. And in the beginning, when they were starting up blood drives again, it was a way to find out something about your exposure or something. And I finally it's come up. But I have the feeling that at this point it's only going to tell me that I'm S positive.
Yeah.
Yeah. but yeah, I, on a quick glance, I can't really tell. And then I was just going to go back. We didn't finish the last part of Laura's question. If a person had an asymptomatic COVID exposure, what does that imply about their need for worry on exposure to future COVID, barring significantly more lethal or infectious variants? And I think having an asymptomatic COVID exposure is not going to make you any more or less susceptible than if you'd had a symptomatic COVID exposure or if you'd had maybe no exposure. I think there's just no telling how much you should worry about future COVID variants.
Yeah, agreed. All right, time for some picks. Kathy, what do you have for us?
I picked something that our friend Justin sent to me. It's a wastewater dashboard, and you can put in different locations, and then toward the top under the gray box, you can click for SARS-CoV-2 or monkeypox or influenza A or respiratory syncytial virus, RSV. And for instance, it's for Ann Arbor. The monkeypox is like down at the baseline. But influenza A, it shows some surveillance. This particular graph is from October 21st to November 11th. So there's a bit of a delay, but it's kind of cool. You can check things out. And then I'm trying to make it through about 150 emails that came up while I was busy the last couple of days. And we got a notification from the Michigan health officer that severe illness with adenovirus infection has been on the uptick here at the University of Michigan. So they talk about how it's usually in the very young or the immunocompromised, but it's been reported recently in our community in several otherwise healthy individuals. So
severe illness, do they specify what severe illness is?
No, they don't.
Adenoviruses can do all sorts of things, right, Kathy?
Yeah. Common respiratory virus typically causes runny nose, sore throat, and fever, but might also include cough and GI symptoms spread by droplets from the nose and throat, contaminated surfaces, no treatment available, no adenovirus vaccine available. So, yeah, just did not know that I was going to open my email and find out that adenovirus was in the news.
I looked at a couple of locales here, and looks like SARS-CoV-2 is dropping in wastewater. In most, there's no monkeypox in a few places that I've looked at. Newark, New Jersey, Concord, New Hampshire. There's no more. Rochester, Minnesota. That's very interesting. Okay, cool. Very cool. Rich, what do you have for us?
So this is not unusual. It was yesterday that the first mission of the Artemis program was launched by NASA. And I just can't resist highlighting this with actually I picked three things because I wanted to sort of fill in my own understanding of what this mission was about. On a previous TWiV, we've talked about our Artemis before, and on a previous TWiV, I think there were a couple of times I picked this, but I picked the whole program, which has as its ultimate goal essentially colonizing the moon, including establishing an orbiting platform around the moon, essentially a space station where you can park ships that will come and go from the surface and potentially serve to launch to other more remote locations. So Artemis 1 is basically testing some of the hardware that will be used for this whole thing in an unmanned mission. It's been under development for almost 20 years, including the Space Launch System, which is a big rocket with a couple of boosters and in this particular case, a capsule and a second stage on top of that. It's using primarily Well, it's basically capitalizing on stuff that's been used, that's been learned from previous missions. So the shuttle mission contributes the solid rocket boosters, the engines, and the fuel storage is more or less copied from the space shuttle missions, and of course the capsule designs and that kind of stuff are developments that advance on those technologies. This mission is unmanned. It's got 3 mannequins in the capsule, and it took off yesterday and is headed for the moon. Cool. And it's go to the moon and orbit for about a month, and then come back and collect data all the way. So this is the first such mission. And I picked. Actually, it turns out that NASA, they have a regular Artemis site and an Artemis 1 mission site. The link I picked is sort of a special site that they put together when they know that a lot of people are going to be watching. And it's got a nice cutaway of the launch system, and it's got a nice video that summarizes the mission. And I also picked a YouTube video of the launch itself and another page from NASA that is a map, a diagram of the mission. So I think it's cool.
Very cool.
I like it.
Yeah, it's very cool. Love it.
So I want to know, do the mannequins have that crash test dummy symbol on them?
I can't see. I do not know. But the mannequins are recording a lot of data. You know, when this thing re-enters, apparently it's coming in faster into the Earth's atmosphere than previous missions have. They get up almost to 10 Gs.
Wow.
They're over 9 Gs. So, you know, I don't know how much humans can stand for how long, but that's 10 Gs would be interesting.
Interesting. I bet some roller coasters do that. Is that possible?
I don't know. We'll get some space nut out there to write in and tell us gravity experiences and how serious this is and where we might come close.
Well, we have Chiara Mingarelli coming onto TWiV. She's going to talk about, she's an astrophysicist.
Joe, I don't know how much she'll know about re-entry in a space capsule, but
maybe not.
Hey, Kate, if you're listening.
I don't think Kate's listening,
right.
but
probably not. Who knows?
All right. I have a food channel on YouTube, which I really like. It's called Pasta Grammar. It is Eva and Harper. So Eva is an Italian lady and Harper's an American. And they teach you how to cook, basically. She is completely Italian, born in Italian. She has a huge accent, which is just charming as hell. And she cooks, and he's talking to her, and he's filming it. It's nicely produced, and they have dishes from all over Italy. It's quite interesting, and she talks about them, and then they try them, and they have a lovely interaction. It's just wonderful. I think it's so good, and so if you want to learn how to do it, they have all the recipes down there, but just watching them is very entertaining, I find. It's just great. I think they live in Arizona, and periodically they go to Italy and travel around and stuff. Some of the things here that are very interesting. So, you know, there's a famous chef by the name of Gordon Ramsay, right? He made a number of months ago a video on how to make carbonara, pasta carbonara, right? So she critiques it. And she said, "This is garbage. This is not carbonara." It's just great. You have to watch that one. So she reacts to a lot of supposed chefs making different Italian dishes. It's very good. So really entertaining, good recipes too. And I just think it's wonderful. The two of them have a great rapport, and you can learn how to make all these things.
I'm going to have a look at this one on 5 that has 5 eggplant recipes.
Yes, that's very interesting.
5 recipes that will change your mind about eggplant. Because I really like eggplant, and I only I have one, you know, sort of, I bootstrap my way through eggplant Parmesan. And as I was making this the other night, I was thinking to myself, man, I got to try a rollatini sometime. But
Yeah.
now we got even more ideas here.
Yeah, eggplant. Then there's another one on zucchini. She says a lot of people don't like zucchini, so she gives 5 recipes that.
Oh, this looks great. Thanks.
And
Vincent.
so
Mm-hmm.
it is, cooking is science, so you could always consider that. But it's very, very well done. He does a great job on the video. He edits them all himself, and they're well done. I have a listener pick from Rich, who writes, so much we can learn from watching a virus in action. And this is a video from Duke today. Watch a virus in the moments right before it attacks. Duke researchers have captured the first real-time footage of viruses on the move right before they attack a cell. I don't know if I want to.
Oh, so I watched it.
Yeah.
And it does come on with audio, but I was a little disappointed because it is only right before it attacks.
Yeah.
you don't get the what happens, but
It's a minute.
You see the
cool.
virus particles zipping around.
Yeah, virus,
Yeah.
and they trace it. Yeah, they're tracing the virus part.
and the shapes of the nearby cells that I think are must be false colored, but they're really nice.
All right, thanks, Rich.
So-
Also, we have one from Justin, probably the same Justin that you have
Yes.
yours, right?
Yeah.
I recently found this excellent review article about RSV, respiratory syncytial virus. It includes discussions of the virology, immunology of RSV, as well as a great section on RSV vaccines looming on the horizon. In addition, there's a good discussion about what might plausibly be driving the recent large wave of RSV-related hospitalizations. This is a very interesting topic because there's been a lot of RSV, especially in kids lately, and people are saying, well, is that just because of sheltering, lockdown, or is there something else going on? I don't know that there's any evidence for something else, but...
So the article has a nice table in here about, or a nice chart, it's a table, about RSV vaccine and mRNA antibody, and MAB, what?
Monoclonal antibody?
Monoclonal antibody snapshot, okay, that has preclinical phase 1, 2, 3 market approved vaccines. So there's an AstraZeneca, that's immunoprophylaxis. So they got some monoclonal antibodies. There's looks like a couple of virus vectored vaccines, one from MVA, a pox virus, and the other from an adenovirus that are in phase 3 trials. There's Moderna has an RNA vaccine in phase 2 trials. There are protein vaccines from GlaxoSmithKline and Pfizer in phase 3. And there's some live attenuated or chimeric vaccines in phase 2. So there's a lot of stuff on the horizon. Cool. Thank you very much. I'm interested to see that.
Yeah, they have a section on the history of the Lot 100 catastrophe.
Mm-hmm.
Yeah.
Is that the enhanced disease catastrophe?
Yeah, I think it must be. I'm trying to read it without.
This looks like a really good article.
Yes.
All right.
All right,
Thanks, Justin.
That is TWiV 956. Show notes are at microbe.tv/TWiV. Send your questions, comments, pics to TWiV at microbe.tv. If you like our work, please support us. You can go to microbe.tv/contribute. There are a number of ways there, including smile.amazon.com. It's there, and you can have them direct the part of your purchases to Microbe TV. You have to tell them ahead of time. You have to specify your charity. And we're there. Some people say they can't find us, but we're there. I found it. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.